students, welcome to Biomix Sub. In this today's video, we will discuss about the protein targeting in case of peroxisome. So this peroxisome which is present, it is a single membrane bound. It is a single membrane bound cell organelles. So what is the main function of this peroxisome? The main function is that it will help in detoxification of the cell. So in the cell, there are hydrogen peroxides which are formed due to some of the reaction. If this peroxisome will not be degraded, then it will be toxic to the cell. So this peroxisome which is present, it is having a enzyme which is known as catalase with it. This catalase help in converting this hydrogen peroxide into H2O and O2. Except this catalase, it is also having one enzyme which is known as urate oxidase. Okay. This was the first thing which is done by this enzyme. The second thing is the beta oxidation. So where this beta oxidation is taking place? It is taking place in the mitochondria as well as in peroxisome. So there are long chain of fatty acids which are formed. This long chain of fatty acid, when there are more in amount, they needs to be converted into their small subunits. This long chain fatty acids which are formed, they are formed from 2 carbon containing compound, okay, which is known as acetyl coenzyme A. So due to beta oxidation, the conversion of this long chain fatty acid will be to its precursor, that is a 2 carbon containing compound which is known as acetyl coenzyme A. In case of mammals, the beta oxidation is taking place in the mitochondria as well as peroxisome. Okay, but in case of plants or yeast, the beta oxidation is taking place in the peroxisome. Okay, and this peroxisome which is present, mainly it is present in the liver cell, and kidney cell, because here in case of the liver cell and the kidney cell, here the detoxification is taking place. So for detoxification, there are certain enzymes which are required, which are produced from this peroxisome. Okay, so now let us understand if it is. In this video, we will discuss about the protein targeting in peroxisome. So if a protein is present, it will have a N-terminal end and a C-terminal end, and for the targeting of this protein into some organelles, any cell organelles, it will have some specific code with it, which is known as signal sequence. And for targeting a peroxisome, the signal sequence will be present in both N-terminal end as well as in the C-terminal end. If it is present in the C-terminal end, it is made up of 3 amino acid, which is known as serine, leucine, and lysine and leucine. If it is present in the N-terminal end, it is having 9 amino acid with it. Okay, so the first amino acid in case of N-terminal will be arginine and lysine, and the second amino acid will be leucine, valine and isoleucine. Next, the 5 amino acid which will be present, it can be any of the amino acid, but it cannot be proline. The next will be histidine and glutamine. The next amino acid will be leucine and alanine. Okay, so according to this, the signal sequence can be targeted in 2 ways, like from PTS1 and PTS2. So the signal sequence which are present in the C-terminal end, it is done by the PTS1. If the signal sequence is present in the N-terminal end, it is targeted through PTS2. Okay, so this PTS1 is it is the most commonly used targeting for the protein sequences. Okay, so what happens if the protein is present in the cytoplasm? It will be in the folded form. It will help in the folding, but here the folding is being prevented by some other proteins. Okay, so here we will, with the help of this diagram, we will understand here. Here the protein is having a signal sequence in the C-terminal end. If it is having a signal sequence in the C-terminal end, here the targeting will take place by PTS1. Okay, so for receiving the signal sequence, it is having a receptor which is known as PEX5. PEX5 is a receptor which will bound this signal sequence with it, and it will help in transferring this protein into the membrane bound membrane bound protein which is known as PEX14. Here the PEX14 is present, which is a transmembrane bound protein. Okay, or transmembrane bound receptor. This is a soluble PEX5 which is present, it is a soluble receptor, and PEX14 which is present, it is a membrane bound receptor. Okay, the after reaching here, this will help in targeting this protein into the matrix protein. Okay, so here there are other, so here the PEX5 which is present, it is being utilized now. It does not have any work here, so it needs to be recycled. So the recycling is done by ubiquitination. Okay, so this ubiquitination is done by another transmembrane protein which are known as PEX2, PEX12 and PEX10. They help in ubiquitinating this protein, this PEX5 protein. Okay, after this there are some other proteins like PEX6 and PEX1 which will do the ATP hydrolysis, and it will help in recycling of the PEX5 receptor. Okay, the next thing is when we will target, when the proteins are present in the N-terminal end, the receptor will be, if the protein are in the N-terminal, and it will be targeted through PTS2. Okay, it will be targeted through PTS2, and for that the receptor which will be present for receiving the protein sequence is the PEX7. Okay, here the PEX7 will work, and the soluble cytoplasmic soluble protein which was PEX5, here it will be PEX19. Okay, and the membrane bound receptor which was present here, it will be PEX3 and PEX16 in case of PTS2. Okay, now we will discuss how this is being regulated, how the division as well as biogenesis of the peroxisomal membrane is taking place. So here there was a precursor membrane derived from the ER which will give rise to peroxisome. Okay, next the peroxisomal membrane protein comes and bind with it, which is known as PEX3. Along with it comes the PEX3 and PEX16. Okay, these are the receptors, transmembrane proteins. So these things will be placed in this precursor membrane derived from the ER, and this all put together they are known as PMP70, which is known as peroxisomal membrane protein 70. Okay, after that the PST1 bearing matrix proteins as well as PST2 bearing matrix protein will come and bind, which is known as PEX7 and PEX14. Next year will come and bind the PEX14. Next year will be PEX10, PEX12 and PEX2. Next PEX1, PEX6 and PEX15 will come and bind in this region. Okay, due to which there will be mature peroxisome. Okay, after the maturation of this peroxisome there will be division. So this division is being taken by PEX11. Due to PEX11 the division will take place. So what happens if this PEX11 is present in more amount? If PEX11 is present in more amount, that means there will be more peroxisome will be high. If there is PEX11 which is present, if it is in less amount, that means the peroxisome will be less in number. Okay, what if there will be mutation in this proteins? If the mutation is in this protein like PEX10, PEX12 or PEX2, if there will be mutation, the ubiquitination of this PEX7 or PEX5 cannot take place. Okay, due to this there will be no recycling. So in this case there will be low amount of peroxisome formed. So if low amount of peroxisome is formed, the catalase enzyme which was helping in detoxification, it cannot be formed, due to which there will be toxic as well as some of the disease in case of organisms. Okay, so here the disease which is due to less amount of peroxisome is, or we can tell mutation in this PEX10, PEX12 and PEX2 is Jelly-Warger disease.